Hey everybody, so I actually figured out what the problem was with the laggy, crappy drawing, and it turns out it was the software I was using. So if you ever want to make one of these, don't use Paintbrush for Mac. This is Autodesk Sketchbook Express, and it seems to be working much better. See, look, I can even do this. Ta-da! And there's no lag when I write or draw. And infinite undos. Hooray! Okay, so today I'm going to be talking to you about the neuromuscular junction, and because I have awesome controls, I can do all kinds of stuff with this. So, neuromuscular junction basics. This is going to resemble very strongly the drawing I made in class if you are in my 231 class. So feel free to follow along with whatever notes you have. So the first part of this is the neuro part, because we have neuromuscular. So that implies that there must be a neuron present. So here we have the axon, and the widened end of it is the synaptic knob, or synaptic end bulb, or axon terminus. There are multiple acceptable terminologies for this thingy. And then inside of the synaptic knob, we've got an assortment, oops, that one's not closed, of vesicles containing neurotransmitters. So let's label that. And then let's pick a color for our neurotransmitter. I'm going to make acetylcholine be green. So inside of each of my little vesicles, I'm going to draw some green dots to represent my acetylcholine molecules. So up here, let's draw green equals acetylcholine. And I'm going to abbreviate that ACH. Okay, so we've taken care of most of the neuron part, but we haven't yet addressed the muscular component, so let's do that. I'm actually going to leave my color editor over here for a little bit so I can have access to all the good colors. Okay, so muscular, I'm going to draw in pink. And I need to make myself a sarcolemma. So this is going to be one muscle cell being innervated by one motor neuron. So we can assume that there are other muscles in this motor unit. This is just one muscle fiber. And notice I'm drawing folds here. That is because the sarcolemma at the motor end plate, which is the name for this particular spot, has what are called junctional folds. So instead of having a flat membrane here, there are deep crevices. And this is going to create more surface area on which the cell can put receptors for acetylcholine. So let's label some stuff here. So this whole piece is the motor end plate. The pink stuff is the sarcolemma. And these little guys are junctional folds. So junctional folds. Great. Okay, so now we have our two cells more or less set up. So what we're going to need to do is figure out how I can get excitation in the neuron to jump over this gap here to become excitation in the sarcolemma. And let's do some other labeling while we're at it. So this is the synaptic cleft. And I'm emphasizing the synaptic cleft because this cell membrane and this cell membrane are not touching. You need space between the two cells for acetylcholine to diffuse across. Now, let's say that an action potential comes down the axon. So a command has been issued from the brain to activate this particular muscle fiber, and the action potential is going to come down this direction. So electrical excitation is traveling this way. At the area of the synaptic knob, we have a couple different kinds of ion channels. So you know from action potential videos and discussions that this excitation change is a result of the flux of sodium ions from outside of the axon to inside of it, and then with inside of it, the ions moving down the axon to excite the next area of membrane. But that by itself can't result in release of neurotransmitter. Instead, what you need are voltage-gated calcium channels. So let's label those. So remember that voltage-gated means these are only open at a particular voltage, and in the case of voltage-gated calcium channels, that voltage happens to be the same voltage as an action potential achieves. So the action potential ending up here is going to act on these channels to make them pop open. And if that occurs, calcium can come in. So let's draw some calcium diffusing through these now open channels into the synaptic knob. Now, one mistake that I've noticed some of my students making is assuming that this calcium and these arrows mean that there's calcium inside the vesicles. That is not true. The vesicles contain acetylcholine, as you can see. The reason that we need the influx of calcium is because the influx of calcium through a bunch of intracellular processes that I'm not going to ask you about cause the exocytosis of vesicles of acetylcholine. So what I'm going to do now is draw vesicular fusion in acetylcholine release. So let's grab a red. So now I'm going to have a couple of my vesicles coming down here and fusing with my membrane. And that results in what was once inside the cell, inside of a vesicle, now it's outside. So we can release our acetylcholine into the synaptic cleft. So here we go. Acetylcholine is now in the synaptic cleft. And that's great. However, it's not good enough. So neurotransmitters have a lock and key relationship with their receptors. Remember, a receptor is a membrane-bound protein that binds to something from outside the cell and causes metabolic changes inside the cell. Right now, our motor end plate is devoid of receptors, so I haven't blessed my sarcolemma and the junctional folds with any way to communicate with this neuron. So let's fill that gap in now. So I'm going to have my receptors be purple. And just for speeding things up purposes, let's make my nib bigger so I can draw these more efficiently. So these are ion channels that only open when acetylcholine is bound to them. So really they're more like a tube, similar to how I drew the calcium channels, but just so I don't waste a bunch of your time making you watch me meticulously draw very complex ion channels, I'm just going to represent them here as little purple hashes, okay? So each of these is a ligand-gated sodium channel. And remember from the other video that ligand-gated channels are channels that are only open when a particular chemical is bound to them. So in this case, that chemical is acetylcholine, and when acetylcholine binds to these receptors, the receptor opens up to let sodium into the sarcolemma. So now I have a robust population of acetylcholine receptors, and let's label those too. And let's also write what they are. Oh, there we go. All right, so let's have our acetylcholine bind to our receptor and see what happens after that. So now we have acetylcholine diffusing in here and binding to the receptor of interest. And I'm not going to fill every single receptor with acetylcholine because acetylcholine binds and then the receptor opens for a little bit and lets go and then binds another unit of acetylcholine and so forth until the acetylcholine from the cleft is exhausted. So if I tell you that the acetylcholine receptor is a ligand-gated sodium channel, and we know that ligand-gated ion channels only open when their favorite ligand is bound, if we have bound acetylcholine, that means these guys are going to let sodium into the sarcolemma. So let's add sodium to the mix. So sodium that used to be out here is now going to enter by passing through, oops, I missed that one a little bit, passing through the acetylcholine receptors. So remember, ordinarily there's lots of sodium outside and lots of potassium inside. And there's more positive charge outside of a muscle cell than there is inside. So we have a polar sarcolemma at rest. If I depolarize it locally, I am making a small patch of the membrane more positive on the inside than usual. So this is accomplished by acetylcholine binding to these receptors and letting sodium cations into the cell. But what happens next? Well, these ligand-gated ion channels are neighbors with, let's make it a different shade of purple, shall we? There we go. It's a nice lavender color. These guys are neighbors with some voltage-gated ion channels. So not ligand-gated, but rather channels that open at a particular voltage. So remember, voltage is how we measure the difference in charge across a membrane, and in cells this is expressed in millivolts. So a excitable cell at rest sits at about -70 millivolts, and if you let more positive charge into that cell, it's going to be depolarized, so getting closer to 0, which is no charge difference across the membrane. So let's label our little guys here. We've got, these are going to be voltage gated sodium channels. So if I've just let a bunch of sodium in here, and there's very little sodium in the sarcoplasm over here, that sodium is going to rapidly diffuse to areas where there is less sodium, and that causes a change in the membrane potential wherever the sodium goes. Turns out that's good enough for the voltage-gated sodium channels to open and again let in sodium. So let's watch this process in action. So sodium, represented here by the arrows, is going to diffuse over to an area where there's less sodium. That depolarizes the membrane enough that these voltage-gated sodium channels open, and more sodium comes in. And then that sodium is going to go over here, and these channels are going to open, resulting in the influx of even more sodium. And that process continues, so these channels cause the opening of these channels, cause the opening of these channels, such that the order in which sodium enters the cell goes from closer to the motor end plate to further down the sarcolemma. And that is the action potential in the sarcolemma. So let's go back to our action potential color. So the action potential in the sarcolemma proceeds in this direction, and in this direction is the T-tubule. And the goal here is to get the action potential to go to the T-tubule, because that is how calcium ends up being released from the sarcoplasmic reticulum. So in another video, not this one, I will show you what happens once the action potential gets to the T-tubule, how calcium is released and restored in the sarcoplasmic reticulum, and what effect that has on the sarcomere. So, thanks for your attention. I hope you find this video useful. See you in the next one.